Hello, everyone joining us. Thank you so much for being here. My name is Karen Schroeder. I'm a postdoc at Columbia's Zuckerman Institute and one of the organizers of a Neuro Launchpad Trainee Talk series. Welcome to the second ever Neuro Launchpad Talk session, a platform that's by and for trainees to share and discuss their science. So this season we have a slate of 10 sessions scheduled through January, which you can check out on our website, neurolaunchpad.com, or on our Twitter. And so far we're having a lot of fun, so we hope to continue this event into future seasons. So today our theme is cellular and molecular mechanisms in development and repair. And we have two great talks coming up. If you have any questions for the speakers, please submit them in the Ask a Question box, and we'll have time for Q&A after each talk. So I'm going to go and introduce our moderator, Professor Alex Jaworski, who's going to take it from here. He's an assistant professor of brain science at Brown. And we also have a wonderful co-moderator, Lena Ali, who is a postdoc at Harvard in the lab of Gorda Fischel. So she'll be handling questions from the audience. And Alex, we seem to have lost Alex's video.
Oh. So people cannot see me? My camera seems to be on.
We can see you.
Okay, and my sound is okay too?
Your sound is okay.
All right.
Everything's working.
So thank you, Karen. I think it's really great to be part of this as a moderator. It's a great idea to have this type of forum. So without further ado, it's my pleasure to introduce our first speaker, Yasuhiro Itoh. Yasu is currently a postdoc at Harvard in Jeffrey Macrophages lab, and where he's studying corticospinal connectivity. He earned his bachelor in engineering from the University of Tokyo. He also completed a PhD at the University of Tokyo, working with Yukiko Goto on neuronal fate commitment and migration in the mouse neocortex. And so today he's going to tell us about his work in Jeffrey Macrophages lab. He's been there for 6 years, and I'm very excited to hear about your work.
Thank you for the kind introduction, Alex. And let me share my screen.
Something seems to have gone a bit wrong there. Let's get him back.
Yeah, I'm sorry. I was trying to share my screen, and somehow I got deleted.
No worries.
Do you see my screen, or do you see me talking?
Right now I see you. Oh,
okay.
Great. Now I see your screen.
Hey, is it working fine?
Yes.
Okay, great. Well, thank you, Alex, for the kind introduction. And I would like to thank the organizers for this great opportunity. And today, I'm going to tell you about two stories about descending axon projection coming from the cortex down to the brainstem and spinal cord, as you can see in this. Using the first 15 minutes, I'm going to tell you about the identification of the proteoglycan lumican for the first time in axon development. And I'm further going to tell you that lumican regulates the specificity of branching of two populations of corticospinal neurons in a non-cell autonomous manner. Then I will touch upon lumican's interesting relevance to the neurodegenerative disease ALS and the evolution of corticospinal circuit. Using the last 5 minutes, I'm going to briefly tell you about some new approaches our lab reported last year to look in depth and quantitatively at the subcellular molecular machinery in growth cones of subtype-specific neurons in vivo. And then I'm going to tell you briefly about my application of the approaches to subcerebral projection neurons with special attention to the functions of the key central transcriptional regulator CTIP2 in the cerebral cortex. Start with the first part, which is equally contributed by our former postdoc, Vibhusani. Our brain has immense capacity for perception, cognition, and internal thoughts, but its ultimate output to interact with the external environment is motor movement. A mammalian brain has a population of cortical projection neurons that send direct projection to the brainstem and spinal cord, and they enable voluntary and skilled movements such as speech and precise finger movements. Corticospinal neurons are somatotypically organized in humans. We all know that they are laid out from legs medially to arms faced laterally. And same is true in rodents, where rostral corticospinals are bulbocervical projecting and medial corticospinals are thought to be thoracolumbar projecting. So I tell you something different we have discovered later in my talk, but I'm going to call them SCS and medial for now. And the question we have been trying to address is how this segmentally specific axon targeting is achieved during development. Though this is the general layout in textbooks, we first wanted to look at this somatotypy with more precision so that we could isolate bulbocervical and thoracolumbar projecting corticospinals. To make long story short, we use both retrograde and anterograde labelings. In the retrograde labeling, you see a defined thoracolumbar area in yellow and a larger bulbocervical area in green, while in the AV anterograde labeling, you see green axons from rostral sensory motor cortex mostly terminate mostly in the cervical segment, and the red axons from caudal medial extend all the way down to the lumbar cord. So we have validated that this is their spatial localization. So and then we isolated cortex from each area. We focus on the first postnatal week when corticospinals are entering spinal cord, stopping or keep going, and then actively collateralizing. We have identified a number of differentially expressed genes, but here are two exemplars, KH-like14 and CRIM1. So bulbocervical specific KH-like14 is expressed laterally, and thoracolumbar specific CRIM1 is expressed medially. And essentially, they are non-overlapping populations. Although I don't have time to tell you the experiments, we have found in this work that KH and CRIM regulate axon extension down to specific spinal segments. And in the rest of my, the first part of my talk, I'm going to tell you that lemmican is expressed by bulbocervical corticospinals and how lemmican controls axonal collateralization in the cervical spinal cord. At first, in a simple scheme like this one. So we got interested in lemmican because of its expression profile, showing bulbar-specific expression, and it goes up over time. First, we tested lemmican expression in vivo, and we found that it's very specifically expressed in rostral layer 5, both at the RNA and protein levels. So lemmican is not expressed medially, corresponding to the microarray result. And for the aficionados in the audience, lemmican is expressed only by subcerebral projection neurons in layer 5, but not by callosal neurons. Its expression peaks around P10, and it goes down by P14. It's also expressed in meninges, and all the same signals go away in lemmican knockout. Then what do we know about lemmican? So lemmican is an ECM protein. It can exist as a proteoglycan or KSPG, and proteoglycans in general restrict axon outgrowth. Except a recent study published by Wieland Hartner's group, the role of lemmican in CNS development remains unexplored. So we wanted to know what this molecule is doing. And as I said, proteoglycans are normally secreted and inhibit axon growth. So we thought that lemmican is likely not a cell autonomous axon growth regulator. but it raised an interesting hypothesis of a secreted molecule that keeps other populations out. And I'm going to tell you that's how it works. But that said, of course, as a baseline, we wanted to ask if there were massive effects of Lumican knockout on early corticospinal development. And I'm not showing you the primary results here, but we did a series of experiments in that Lumican knockout can show normal specification, normal subtype development, normal axon entry into the spinal cord, and normal axon extension within the spinal cord. So now the baseline is set. So then, to test the hypothesis that Lumican is keeping other population out, we injected a trace of dye-BDA into cord medial corticospinals that do not express Lumican, and then traced axon branching in the cervical cord. So here you see the BDA injection site in the cortex, and here there's a dramatic difference in the total amount of cervical axon collaterals in Lumican knockout. So with careful normalization to the number of CST axons in the cord, so there is a substantial and significant increase in the collateral density in Lumican knockout, indicating that Lumican is non-serotonously suppressing axon collateralization in the spinal cord. We tested the effect of Lumican overexpression. So in this experiment, we injected AAV laterally to overexpress Lumican, and then injected BDA cord medially. We verified that there's no overlap between these two injections. As you can see in these images, so Lumican overexpression caused drastic reduction of axon collaterals in the cervical cord, and the quantification demonstrates very efficient suppression of axon collaterals by Lumican overexpression. To summarize the results so far, we found that Lumican is expressed by bulbous cervical corticospinals, and Lumican is suppressing axon collaterals from cord medially located corticospinals in a non-serotoninous manner. So now some of you might have picked up the fact that I was using CSMBC or bulbous cervical for the rostral corticospinals, but referring to medial ones as the same medial. So this is because we didn't want to mislead you. So we have discovered that there's another bulbous cervical projecting population interspersed within this medial population. And some of you might know emerging literature in primates that these are evolutionarily older populations. So we renamed them, and importantly, all of these corticospinals innervate cervical segment. And quite interestingly, this medially located bulbous cervical corticospinals in blue don't express Lumican. So then we wondered which, or maybe both, of these two medial corticospinals are regulated by Lumican. So using conventional genetic and viral tools, we can dissect these two populations, and I'm going to tell you some very interesting results we found. But to make the very long story short, we have discovered that Lumican suppresses axon collaterals of both medial corticospinals. And to save time, let me show you here only the results of this red thoracolumbar projecting corticospinals leveled by CRIN1 Glial knock-in system. This intersectional genetic approach enables us to specifically label thoracolumbar projecting corticospinals. So these are axial sections of cervical spinal cord, and compared to white type, there was a substantial increase in collateral density in Lumican knockout, as shown in these images, and in quantification, which is throughout the cervical cord, indicating that Lumican non-serotonously suppresses axon collateralization of thoracolumbar projecting corticospinals. Finally, let me show you some interesting results about ALS. So it causes degeneration of low motor neurons and upper corticospinal neurons. A previous study identified the same amino acid substitution only in ALS patient cohort, but not in control cohort. We mutated this conserved leucine residue in mouse Lumican to see if this substitution changes any of Lumican function. when lemmican was overexpressed by cortical primary neurons in vitro, lemmican protein was very efficiently secreted into the medium. And quite interestingly, this secretion, the amount of secreted lemmican mutant was decreased compared to lemmican wild type. So because lemmican is expressed only during the first 2 postnatal weeks of corticospinals in mice, so we speculate that perhaps if corticospinal circuitry is made subtly abnormally during development, there might be later-aged degeneration. And to summarize the lemmican part, I first showed you anatomical and molecular heterogeneity of corticospinal neurons, and then showed you that lemmican is expressed by dorsolateral bulbar cervicals, and lemmican non-serotonously suppresses normal quantization of 2 corticospinal subpopulations located medially, as shown in this working model. And importantly, this is a segmental level specific mechanism, and lemmican mediates axon-axon interaction between corticospinal subpopulations. And we also showed lemmican's potential relevance to ALS. And finally, this mechanism might give us an important insight into how the corticospinal circuitry has evolved. And let me use the next 2 slides to elaborate a bit further in this direction. So to address the question, how motor cortex emerged and has during evolution, John Kars and his colleagues have done very nice anatomical and physiological investigations across mammalian species. They found that distinct motor cortex emerged about 100 million years ago in early placenta mammals, and that through mammalian evolution, motor cortex has expanded dorsally and ventrally by adding more motor areas. In mice, lemmican is expressed by evolutionarily newer dorsolateral corticospinals, and in developing marmoset cortex, lemmican expression by corticospinals is nicely conserved across species, and it appears expanded ventrally, consistent with the notion that lemmican is expressed by evolutionarily newer corticospinals. And as you all know, a human brain is much bigger than a rat brain by about 1,000-fold. But in contrast, human spinal cord shares most of the anatomical features with rat spinal cord, and its size has expanded by only 40-fold. So of course, we still need to know if the number of corticospinals in a human brain has increased proportionally. But this imbalanced evolutionary expansion poses us a question: how newly added corticospinals or any kind of neurons integrate into pre-existing circuit during evolution? So our findings suggest that lemmican and related molecules might play key roles in enabling evolutionarily newer neurons to innovate by actively suppressing evolutionarily older counterpart. Okay, using the last 5 minutes, I'll tell you about some very exciting work in progress on the growth hormone biology of subcerebral projection neurons in collaboration with Hatch and Ann Engman in the lab. On the left, you see a cortical projection neuron in culture having dendrites and a long axon. The tip of growing axons, called growth cones, play essential roles in guiding axon projection through sensing surrounding attractive and repulsive cues. So amazing jobs by growth cones are well exemplified in this nice movie by Kristen Holt. So guided by the growth cones, these 2 retinotectal axons project straight toward tectum, but once they come close to the tectum, these 2 axons diverge and then target distinct areas in the tectum. Now you see the targeting tectum. And in textbooks, we see typical non-neuronal cells and neurons like this one, highlighting polarized and distinct morphology. But really, diversity and complexity occurs in vivo. This is an in vivo neuron to scale, and the growth cones are far away from its soma. So our lab developed approaches to directly investigate growth cones of a defined circuit in vivo. Based on biochemical approach reported decades ago, our lab recently developed a novel experimental and analytical approaches to subcellularly map RNA and protein molecular machinery of subtype-specific growth cones in parallel to their parent cell bodies, isolated directly in vivo. So basically, in our new approach, we label specific projection neuron subtype by a fluorescent protein, and then isolate fluorescent growth cone particles by using a fluorescent particle sorter, and cell bodies by FACS. So this approach gives us comprehensive RNA. Thanks. This approach gives us comprehensive RNA and protein landscapes in both growth cones and cell bodies, which enables us to elucidate molecular machinery in vivo growth cones. And here we have two major motivations for this work. So first, we want to know the growth cone molecular machinery in subcerebral projection neurons, or SCPN, including corticospinal and corticobrainstem projection neurons. So our subcellular mapping approach was developed using callosal projection neuron, shown dark green on the right, but the growth cone machinery in corticofiber projection neurons remains unknown. And secondly, we would like to investigate how SCPN growth cone molecular machinery changes in abnormally projecting CTRP2 knockout axons. And we anticipate that knowing what kind of growth cone molecular machinery is going wrong in abnormally projecting axons would give us ideas how to reactivate the growth of damaged corticospinal axons for repair. So let me briefly explain about CTRP2 using this slide. So our lab previously identified that transcriptional regulator CTRP2 is expressed by corticofibers, including SCPN in layer 5, and that CTRP2 is critical for both axon growth and fasciculation. Growth and fasciculation for all corticofibers, including corticospinals and broader subcerebrals, as exemplified in those lower panels. Importantly, CTRP2 is critical. CTRP2 has become a central key regulator in cortical development because it's either activated or repressed or cross-repressed by other key regulators. And because of that, I've selected CTRP2 for this first investigation. So far, we have successfully isolated SCPN growth cones and cell bodies from both Y-type and CTRP2 conventional knockouts, as briefly explained here. So we use cortex-specific Glial to label all cortical projection neurons, and then isolate SCPN growth cones from the brainstem and spinal cord, as you can see here. For isolating SCPN cell bodies, we inject retrograde tracer into the peduncle, and then FACS sort SCPN cell bodies the following day. So we are about to complete collecting samples for subsequent analysis, and I would like to share and discuss those exciting results hopefully soon, maybe in some later season of this series. And in the last slide, I would like to thank my colleagues in the lab, especially my mentor, Jeff, for his very generous and insightful support. And I'm happy to take any questions.
Thank you, Yasu. That was a great talk. If you don't mind, I will start with asking you.
Yes, please.
Yeah. So I might have missed it, but do you know that Lumican is supposedly deposited only at the cervical level in the spinal cord? That seems to be your working hypothesis. So
yes, yeah, it's expressed by corticospinals that project down to cervical, but
right.
It's cervical specific.
So do you actually see the protein being deposited selectively in that spinal cord segment? And the other question would be, do you think it's being released from growth cones as these axons elaborate branches there, or from the axon shafts, or what is your idea?
So my idea is at least we don't know. At the moment we don't know. But what I have successfully done is to do Western blotting, taking out some tissue block from cervical segment, and I could detect the protein. And also when I overexpressed lumican, I could stain the protein in the cervical cord. So I think it's trafficked down to the cervical segment, but I don't know how it's really released or exocytosed. Maybe it's activity dependent, maybe it's constitutive. That's still an unknown point.
Okay, we have one question from Jonathan Singh Alvarado, and his question is, the lumican results are really dramatic. Is the effect of lumican only visible at the level of axon collaterals or terminals? This is in relation to Alex's question as well. In the spinal cord, or can you see these influences even within the cortex or the descending pathways prior to the spinal cord?
So in the knockout, we don't see any difference in the extension of CST axons in the knockout. I need to still test that in the overexpression experiment. But so far, as far as I tested, there's no effect on CST, like main axon tract.
But are there any central differences, like within layer 5 of the cortex? Do you see any changes in the cell's morphology or any other type of differences within the cortex?
So within the cortex, I checked layer structures or overall brain structures and other types of projections. But so far I have never found any other phenotypes. What we have found is very specific to corticospinal axon branching.
I have another question. I was wondering, well, I have two questions actually. One is, does lumican have any cell autonomous roles, or does it only act cell non-autonomously? And the other question is whether there is something similar to lumican that works on the other side, like the medial affecting the BC corticospinal axons.
Yeah, so to answer the second one first, so we don't know, but it's a possible, it's interesting possibility. But what I can say is that, so lumican is a member of 18 gene family, but none of these family members are expressed in the other corticospinal populations. But there may be some other similar, that's in the similar job. And to address your first question about cell autonomous functions, so first in the knockout, so when I check the axon projection from the area expressing lumican, I see some modest reduction it's a modest reduction of axon collaterals, which is not statistical, that's not significantly different, but it's modest reduction. And my interpretation is that it could be secondary effect by the increased axon collaterals from the other population. So this is in line with my idea about evolution. So those new evolutionary newer ones are removing previously existing axon collaterals so that they can have some space for making connections. So that's what I have found so far.
Very interesting. Maybe I can ask one last question before we proceed to our next talk. And I was wondering if there are any common targets. So I'm assuming that these corticospinal neurons also send collaterals to other brain regions. And I was wondering if any of the medial versus the BC corticospinal neurons, if they target anything commonly.
So common target means bulbar cervical and thoracolumbar.
Right. It's not within the spinal cord. I mean within the brain.
Oh, okay. So I think they both have some collaterals to some extent in the brainstem. So it's quite interesting to look into brainstem, but we haven't had time to investigate brainstem. But that's interesting. Yeah.
I think we just
thank
got
you.
one more question in the chat, which we
Oh.
probably have time for.
Sorry about that. Okay, so there's one more question, and it's from Kader Ostan. The question is, do you know whether thoracic or lumbar neurons have similar non-autonomous functions over cervical neurons to inhibit their overshooting to caudal areas?
So I'm trying to interpret the comment. The thoracolumbar neurons. Yeah, that's related to your question, and we don't know. It's possible, but yeah, it's interesting to look into.
All right. Well, let's thank Yasu for his amazing talk,
Yeah.
and I think Alex will now introduce Oshri.
Yes. So our next speaker is Oshri Avraham, who is currently a postdoctoral fellow with Valeria Cavalli at WashU, and where she's studying axon regeneration. She trained as a graduate student with Avihu Klar at the Hebrew University in Jerusalem, and where she studied mostly mechanisms of axon guidance, but as I said, in Valeria's lab, regeneration, and that I think is the topic of her presentation today as well.
Correct. So thank you, Alex. Can you hear see my presentation? Okay. Okay. So yeah, so in Valeria Cavalli's lab, we're interested in molecular mechanisms of axonal regeneration after central nervous system injury and peripheral, compared to peripheral nervous system injury. And my work in the lab really focused on non-neuronal cells and the contribution of the environment of the neurons to those mechanisms. And injury and regeneration in the peripheral nervous system compared to central nervous system is very different. For example, if you injure your finger, quite shortly after, you will fully recover. However, this is not, unfortunately, the result after an injury to the brain or to the spinal cord. So this is mainly a result of two mechanisms or factors in the neuron. One is the activation of neuronal pro-intrinsic, neurointrinsic pro-regenerative programs, which show to be very weak or even absent after central nervous system injury. However, decades of research really show the emphasis of many different mechanisms upregulating after peripheral nervous system injury in the neurons to activate efficient regeneration. So another factor that most recently have more emphasis on with many publications in the central and peripheral nervous system is the contribution of non-neuronal cells that are in the neuronal environment and how they inhibit regeneration in central nervous system and how they can promote regeneration after peripheral nervous system. So the model to study differences between peripheral and central nervous system that we use in the lab is a dorsal ganglion. And the dorsal ganglion is have the sensory cell somas, the sensory neuron cell somas outside of the central nervous system. But what's remarkable in this model system is that the same sensory neurons, they're pseudo-unipolar, and every neuron sends one axon that actually bifurcate into two branches. So every sensory neuron sends one branch to the periphery to the sciatic nerve, and one branch it's sent to the central nervous system and enter the spinal cord to ascend to the brain and the dorsal column of the spinal cord. So the same cell, when you injure the periphery of its axon, it will pretty fast regenerate pretty efficiently. However, if you injure the central branch or the spinal cord ascend axon, regeneration will be very limited. And a lot of it is due to the neuronal intrinsic response, as I mentioned. However, if you make a cross-section in the DRG, you can see that the first larger cells in the DRGs are obviously the neurons. However, there's many other cell types surrounding the neurons, with satellite glial cells completely surrounding here the cell somas of the neurons, as well as Schwann cells that myelinate the axons, vasculature cells, and other cells that support the tissue around the neurons. And actually, if we took a mouse that expressed nuclear GFP specifically in all sensory neurons in the DRG here with GFP, completely localized with islet-1, which is a marker for sensory neurons, and we dissociated and sorted those DRGs, and you can see that neurons are only account about 12% of all cells in the DRG. So it gave us motivation to really look at what all the rest of the cells in the microenvironment of the neurons are doing after an injury. And this led us to mainly two questions: how the neural microenvironment really respond to peripheral compared to central injuries, and we really wanted to understand how they contribute or inhibit regeneration in both scenarios. So to do that, we generated a large data set. We're doing a few types of injuries. So for the peripheral injury, we crushed the sciatic nerve. And for central injury, we actually performed two types of injuries. One is crush of the dorsal root, and another injury was dorsal hemisection of the ascending neurons at around T9, T10 of the spinal cord. So in order to look at all cell types in the DRG at once in the same data set, we decided to take the approach of doing single-cell RNA-seq. So we collected DRGs from animals that undergo all different types of injuries. And actually, if anybody's interested, all this data set is publicly available in UCSC genome browser database. And when we sequenced the cells, we actually saw that there's a large group of satellite glial cells, Schwann cells, macrophages, endothelial cells, and mesenchymal cells. And if we separate the cells by the type of injury they undergo, we see big differences, mainly after nerve crush. The clustering is really different than control mice, which suggests that their gene expression is highly changed after nerve crush compared to spinal cord injury. And if we count the genes that are changing in each one of the cell population, we see that there's higher response to peripheral crush compared to a central injury. However, we see that every cell type, neuron obviously respond differently. However, every other cell type in the DRG change their gene expression and respond to the injury in many ways. So I'll just give you an example of few cell types that we looked at their response. And after that, I'll actually focus on the response of satellite glial cells to the different injuries and what we found about them. So we were interested to see that actually parasites and endothelial cells that they build the blood nerve barrier of the peripheral nervous system, that they really respond and change their gene expression after different injuries. For example, if we label those mice with lectin that labels vasculature, you can see that the DRG is highly vasculated. And this architecture of the blood nerve barrier is actually maintained by expression of different tight junction proteins and adherent junctions and gap junctions. And if there's disruption of this junction gene, that could lead to pathologies and disruption of the barrier and could affect neuronal health. So we were looking at our data set at the different injury on those junction genes, and we actually saw many changes occurring in parasite and endothelial cells in the composition of those junction genes. And we're now investigating how those changes might affect the neuronal regeneration capabilities. Another cell type that we were looking at was macrophages. So macrophages are known to be proliferating and contributing to regeneration at the injury site in the sciatic nerve. However, nobody really looked at what's going on in the DRG next to the cell somas. And we see there's many, the responses to the different injuries are quite different. And although we know that macrophages proliferate at the injury site, we also saw upregulation of proliferation markers like K67 and CDK1, specifically after sciatic nerve crush. And we stained DRGs after the different injuries, and we saw really increase in CD68 positive macrophages after peripheral injuries and less after central injuries. And now Ricky in the lab is investigating how, what's the role of macrophages and how this proliferation is affecting regeneration capability of the axons. So now I really wanted to focus on the contribution of the satellite glial cells. So satellite glial cells are glia in the periphery, unique to the periphery. They exist in all ganglion in the body. And the satellite glial cells are actually cells that completely surround the neuronal cell soma. Here you can see example in EM of 3 nuclei of glia, but their membrane is completely surrounding the cell soma of the neuron. And in 3D reconstruction of an EM image, you can see multiple satellite glial cells with the nuclei completely surrounding the neuronal cell soma. So we wanted to know, they're the closest cells to the neurons, and we wanted to see if they have any role or how they can contribute to regeneration after injury. So in our single-cell RNA-seq data, we actually saw that the satellite glial cells was the largest cell population in the DRG. And we characterized them by some known markers like adhering 19 and the potassium channel KIR 4.1. However, if comparing them to the other glia type in the peripheral nervous system, the Schwann cells, we see that they express Schwann cells that express different genes that are not expressed in satellite glial cells like periaxin and NCMAP. However, some genes seem to be shared between the 2 types of glia, like MBP and PLP1. So actually comparing the top expressed genes in satellite glia and Schwann cells, we didn't find many genes in common. However, when we compare satellite glial cell top expressed genes with astrocyte genes, we found much more in common to astrocytes. And major limitation in the field to study satellite glial cells is actually the lack of markers or molecular tools to genetically manipulate those cells and study their biology. So using this data set, I was thinking of maybe I can find a marker or mouse line that I could use to study them. And one marker that we found was fatty acid binding protein 7, or FABP7, also called BLBP, which is highly enriched in the satellite glial cell cluster. And by doing immunohistochemistry of the DRG, you can see that FABP7 is expressed in satellite glial cells around the neurons here, and it's not expressed in the nerve, it's not expressed in Schwann cells. So having this marker, we could find that this mouse actually exists. And by crossing this mouse, the BLBP inducible mouse, with cytoplasmic or nuclear GFP, you can clearly see that it's specifically labeled the satellite glial cells that are around the neurons, and it was not expressed in the nerve in Schwann cells. So that data set also makes us think that satellite glial cells represent a uniform cell population, because we know that sensory neurons in the DRG are different from each other. Are the glial also different from each other? Although FABP7 is a marker that labels all satellite glial cells, by reclustering only satellite glial cells, we found that actually they can be divided to 4 different subtypes. And we found some unique markers that express in each subtype of the satellite glial cells. And one of them was LDH101, which is actually a marker that labels all astrocytes. So we used that mouse, the LDH101, to look if satellite glial cells express LDH101 in all populations. And actually what we found that it support our molecular analysis that in the protein level, it's expressed only in a subpopulation in the DRG and not around all of them. And by labeling some of the neurons with TREC-A, which specific only a certain type of neurons, the nociceptor, small diameter nociceptors, we can see that the satellite glial cells can express around them, or they can be expressed around other types of neurons. Sorry, which suggests that they're not restricted to one type of neurons. They can, the subtypes can express around any type of neuron. So going back to see how the satellite glial cells change their transcriptional response in the different injuries, we divided by injury, and you can appreciate that the satellite glial cells really seem to be dramatically change their clustering, or meaning their gene expression after peripheral injury, and less after spinal cord injury or dorsal root crush. Dorsal root crush also changed, but in a different way. So we said that they might be reprogrammed because they're really highly changing their molecular profile. And actually, after peripheral injury, if you look at the clusters that emerge, one cluster is new clusters, a completely new cluster is emerging only after a peripheral injury. So now we wanted to see what type of genes or pathways are changing after peripheral injury inside of the glial cells. And we found really high enrichment in pathways related to fatty acid metabolism, fatty acid biosynthesis, and PPAR signaling pathways. So PPAR are nuclear receptors that actually their ligands are fatty acids. And when they bind the free fatty acids, they activate a certain transcriptional program. So the enzyme, the committed enzyme in de novo fatty acid synthesis is fatty acid synthase, or FASN. And we wanted to see if it's indeed expressed in satellite glial cells, and we saw high enrichment of its expression in satellite glial cells. And with immunohistochemistry, we also saw fatty acid synthase expressed in satellite glial cells around the neurons. So now we thought if expression of fatty acid synthesis is important in satellite glial cells after peripheral injury, what's going to happen if we now delete fatty acid synthase, specifically in satellite glial cells? So we could do that by the mouse model that we found flocks to a cross to a floxed FASN mouse that we were able to get from the lab of Clay Semenkovich. So you can see that there's no expression of fatty acid synthase in those floxed mice. So those mice didn't seem to have any defect in the morphology of the neurons or in their functional properties. So we wanted to test if we injure those knockout mice, would regeneration be affected? And indeed, this is what we found. So if we injure the nerve and measure the extent of axonal regeneration 3 days after injury, while wild-type animals regenerate pretty efficiently, the knockout animals fail to regenerate as efficiently as the wild-type mice. So looking at the downstream of fatty acid synthase, we found that the subtype of the nuclear receptor PPAR alpha is actually expressed in satellite glial cells here around the neurons. And many of PPAR alpha target genes actually upregulates in satellite glial cell specific after injury, a phenomenon that we didn't see in the neurons. So the question was to follow up, if we activate PPR alpha in the knockout mouse, would it improve their regeneration?
2 minutes.
And indeed, we did that. We fed the knockout and control animals with fenofibrate, which is a PPR alpha agonist. And we found, although we didn't see effect in the wild type animals, the knockout animals' regeneration was completely rescued with fenofibrate treatment. So then we went back to look at the, what's happened in satellite glial cells after sciatic nerve crush compared to the central injuries. And if we saw a highly enrichment in fatty acid synthesis and PPR alpha after peripheral crush, we didn't see that effect at all after central injuries. And furthermore, we saw reduction in PPR alpha signaling after spinal cord injury. So we really wanted to see whether application of the PPR alpha agonist, activation of the PPR alpha pathway would increase regeneration after central injuries. And we did, and indeed we saw that if we treat animals with fenofibrate, the agonist, when usually after central injuries, they're really slow regeneration of the axons. With the treatments, axon seems to be growing pretty well. So actually, fenofibrate is an FDA-approved drug to treat high blood cholesterol, and we wanted to check the feasibility of repurposing maybe this drug to treat humans with central nervous system injuries. So with our collaborators, we collected data from mouse, rat, and human satellite glial cells, and looked at the main pathway that the high differentially regulated nerve genes express. And there we found highly enrichment of metabolic pathways in all species, as well as fatty acid metabolism and expression of PPR alpha signaling pathway, including PPR alpha and its target genes. So I'll just summarize what I showed you today. We found that each cell population in the DRG respond to central and peripheral injury in different ways. Every cell type had a unique response signature, and satellite glial cells actually showed us opposite response between peripheral and central injury. And by modulation of fatty acid synthesis in satellite glial cells, we could improve regeneration after central nervous system injury. I just wanted to mention that our future plans with this data set is actually look at, follow the pipeline that we did with satellite glial cells and look at other pathways and other populations in the DRG, like the HEPA signaling pathway in Schwann cells, the oxytocin signaling pathway in the endothelial cells, and we're now in the lab working on depletion of macrophages and seeing what's their role in the DRG. So with that, I want to thank my mentor, Valeria Cavalli, and the lab, especially Ricky and Eric, that helped with this project and our funding. And if somebody's interested, we have a funded postdoc position in the lab. Thank you.
Great. Thank you so much. That was a great talk. I'm going to take the privilege again to ask the first question. But so correct me if I'm wrong, but presumably the different types of injuries cause different types of gene expression changes in the DRG sensory neurons. And then all the other changes that you see in the DRG resident cells, other than neurons, are a secondary consequence of that. So what do you think is the signal that the neurons produce that changes the behavior of the satellite glia?
Actually, we don't see a lot of changes in neurons after central injuries. So I'm not sure what's following what. I mean, the signal comes from the axons, but it looks like the environment is much more sensitive to the central injury than the. We're gathering now in the lab. It's a different project specifically on neurons, but it seems that their response is really weak, specifically after spinal cord injury. But it's an interesting question what the signals from the neurons to the surrounding environment. Yes.
I have a follow-up on that, actually, very related to Alex's question. If there are no transcriptional changes in the neurons, is there a specific retrograde signal, perhaps, that is coming from the peripheral axons that is perhaps not coming in from the central axons back to the cell body that is then signaling to the satellite glial cells that promotes this whole signaling cascade, which allows for the regeneration? So do you have any evidence of like what, if at all, this retrograde signaling exists, what it could be?
Yes, we haven't looked at that. Yeah, we know that a lot is going on, and it's really well characterized, all the retrograde signaling that going from the injury site into a cell soma, but we never tested after central injury if that is different or not.
And another, I guess, related question to that is, because you showed that the satellite glial cells only encompass the cell body, right? So then why does it differentially affect the peripheral axonal regeneration and not the central one?
Yeah, it's a good question. So we think that the fatty acid synthesis in satellite glial cells somehow contributes to the cell somas, to the neurons to help recover maybe the axon's membrane. But this signal to activate this pathway is not being activated after central injury. So the glia doesn't know that they need to do that after a central injury. So we hope by using those drugs and artificially activate those pathways, we might mimic the expression and the recovery that happens in the periphery.
Very cool. We have a question from Polisi, if hope I'm pronouncing it correct. Is PPAR alpha signaling upregulated in a specific satellite glial cell population subtype?
Yeah, it's a great question. I just recently looked at it. It looks like it's in all subtypes of satellite glial cells. It's not unique to a certain cell type. We're not still sure what's the contribution of each one of the cell types, but there might be other subtle differences between the subtypes.
I noticed in your satellite glial subtype, different subtypes, one of them is the SCN7A that expresses SCN7A, which is a voltage-gated sodium channel. And I was wondering if there are any studies related to what this sodium channel is doing in those glial cells, if it's somehow receiving some type of signal from the neurons, or is it involved in completely a different activation cascade on its own?
Right, it's a great question. I think till now only potassium channel were characterized to express in satellite glial cells, but we see actually in this certain glial subtypes, I saw a lot of sodium channels expressing, and it's really interesting to study how they, respond to signals from the neurons through these channels, but we haven't tested that yet.
If I may ask another question, actually. So there's this well-characterized conditioning lesion paradigm, right, where a peripheral lesion before a central lesion will actually encourage CNS regeneration. Have you done the entire profiling that you've done in that kind of setup? Because there might be gene expression changes in the glia that explain also that type of conditioning lesion effect.
Right, yeah. I think we know that this type of effect, you usually do that ex vivo. You pre-injure the nerve and then you plate the cells and then you see response. But yeah, it's a good idea that to pre-injure the central nervous system and see if plating those neurons, maybe with addition of PPR alpha agonists, and see if regeneration would be increased in that model. It's a good idea.
I actually have another question, which is related to the way you're performing the injuries. I'm actually not very familiar with how these injuries are being performed, but excuse me, because you showed that there are also changes in endothelial cell and pericytes. So when you're doing those injuries, are you also affecting the blood vessels? I'm assuming that the blood vessels are also being affected as a result of the injury procedure itself, right? And not just the nerves, or is there a magical way of only causing nerve damage and not blood vessel damage?
No, well, it's a crush. I mean, you don't cut them, you just crush them. Obviously, it will be affected, but the injury actually is made, distal from the DRG. So it was surprising to us that, yeah, of course, I'm sure the whole blood vasculature is changing in the injury site. However, distal from the injury site, in the DRG, well, the cell somas we didn't expect to see those much of differences, which we need to further investigate.
Very nice. I don't think we have any further questions.
That's perfect. We're right on time. Thank you so much, everyone, for being here. Thank you to our special guests, Alex and Lena, and our speakers. This was great. Thank you. We'll see everybody next week.